Welcome everybody. So I'm Dr. Nicola Knight, I'm the PSDI coordinator, and I'm here today to facilitate this webinar, the third in our series. So today we're going to be hearing about experimental data capture, and we're going to be having some examples of producing publish-ready data from processing and analysis. And we're going to be doing a couple of examples, and then there'll be the opportunity for you to put your view forward in terms of what examples you'd like to hear. So this is going to be presented today by Abraham Nieva de la Hidalga from Cardiff University and Catalysis Hub. Over to you, Abraham.
Okay, thank you, Nicola. Thanks for the introduction and welcome today to this webinar. Let's get started. So the first thing is I wanted to talk about what we are doing with the PSDI Pathfinder related to data capture. And in reality, how this data capture relates to reproducibility and eventually replication of experimental results. So we will talk about these topics here, the speed up of the processing and analysis, how we plan this to help us reduce the time spent in documenting and publishing datasets. These are automated approaches based on Python and large libraries. We have already some examples that you can test and use. If you like, the links are also in the Google Docs document. And we are at the moment still working on the Galaxy version. And well, just as a motivation, we know that in the current context, the scientists produce large quantities of data, and this is only expected to grow. So the example of Diamond 2 that will provide new beamlines with more advanced features and will produce larger data sets with more better definition. So and faster times of producing these results. These are one of the drivers of this. Also, the many proposals that we've seen in literature for higher throughput analysis reactors, like the one in the image. We had experience of that in the Catalysis hub with Xiaojun. And so, and the idea of reproducibility and replicability is this of going up in the scale of this from having mostly research which the researchers can easily run on their computers and repeat n times, to having really reproducible examples which other scientists can take the data and produce equivalent consistent results from your data, and then move on to replicability eventually, so that other researchers can take not only the data, but the methods and apply it in different contexts. So this would be eventually called reusability, so which links nicely to the FAIR data principles. And we have in this context, we would target capturing data in every stage of the research process, from the collection to the publishing of results. And you know that at each stage, the types of metadata produced along with the data it keeps on growing. So it's an additive process. And especially the last part, when we are publishing, this is mainly a manual process where the researcher has to document their data sets before publishing them in some repository. We already proved that many of these tasks can be automated. For instance, we created this SAINS analysis workflow in which we were able to really reduce the time of processing a large data set of more than 3,500 groups from days to some hours, in some cases even really fast speed up when compared to the normal processing that requires these tools that everybody knows, Artemis, Athena, Dawn. They are very good tools. They cater to all the needs, current needs of the researchers. They are well documented, but for high throughput experiments, they tend to form this bottleneck because we still depending on this user interface that hides the complexity of the processing and analysis, and also doesn't produce all the metadata that we would want. So our goal is to guarantee reproducibility. So to some extent, the problem is that this could add to the already demanding sets of tasks of the researchers. And that's why we are proposing the creation or adoption of tools which can produce the required metadata during the processing and analysis tasks. And as I said, these tools that we are testing are based on Python and Larch. We have been working with two parallel versions. One which uses Jupyter notebooks, and the other one which uses Galaxy workflows. The Jupyter-based one is desktop-based, is interactive, and it's ideal for small to medium scale spectra. So about the same as what you use currently down and Athena and Artemis. And the Galaxy base, which doesn't require any setup, is web-based, and it's suitable for very large scale analysis. So, and how we are developing these examples is by looking at published results and trying to reproduce them. So normalization of data, comparing feeds, comparing different signals in 3D plots, plotting data from different with different levels of detail, and highlighting different aspects of catalytic surface, for instance, highlighting the comparison between readings and standards, and the behavior of samples in times, because or linear combination fittings. So at the moment, the 9 examples that we linked in the Google document, they perform all these operations, and they all. But we started to think about how to extract this metadata. So we read the papers, we have written these programs or notebooks, and we try to run some automatic tools to extract the variables, identify the variables declared at each point, and then from this, and then make them into metadata so that these are parameterized and can be repeat or reused. The software becomes reusable for different data sets. And this mapping of the metadata has resulted in this type of documents, which are almost human readable. You can see what figure is this data corresponds to, the links to the publications and data objects with the DOI identifiers, and even the names of the data files and the locations in the computer. So these file mappings are very important for reproducing the results, and it's very time consuming for users to produce them, and especially if we would like some formatting of these data mappings. The operations also need to be codified. So what are the sequence of the operations, which operation goes first, and with which parameters or variables assigned to it. So we will start with a demo, a live demo. So I'm going to pause the presentation for a bit and then go back to my browser, because this demo runs in on the browser. So this is my browser session. Just let me get it here. So use some conventions. This is a Jupyter notebook. I don't know if all of you are familiar. It's just like a web page where you can edit text and code at the same time. The text regions are called markdown cells, and they use there for documenting the process. And the code regions are actually for where the processing of the data happens. All the ones in the examples provided for this webinar, they follow the same pattern. At the top, the name of the task to be performed, then the resources that we need, so the libraries that need to be imported for performing these operations. And then from there, in section 1 is read the data. So first we read the metadata, and then we read the actual data into a structure that holds this data for the rest of the exercise. And then we start running the results. So, for instance, if for running each part of the notebook, you just click on the run button at the top of the toolbar, and then you get immediately an output. So if I do this, clear all outputs, the thing it does is it will go and restart the Jupyter notebook and leave no visible outputs, no results present, and then we can start running the cells one by one, like just by clicking on the run button. When this cell is running, you see a change in the IM that from the empty brackets, it changes to an asterisk between the brackets. That means that this cell is running. When that asterisk changes to a number, then that means that the running has finished. So at the moment, that means that all our libraries have been loaded into memory. And then if we run this first cell that says that this is where we import the data mappings and operations, then we have two subtasks that read first the metadata for the experiment and the metadata for the operations that we need to perform. And then from the next cell, if we run it, it also reads all the data specified in the metadata mappings and collects it into this list of outputs that we call merged results. And then we can go to the next operation, which is, again, we take the variables from the operation, which is specified there. And we, if we run it, then we can see the output. So the reading of these files is the result is immediately below the cell. So this is the first result. And then so just if we just keep on clicking run, we will see the results as they happen. So in this case, there was a normalization of the data, and we see the results of the normalization. If we compare the plots, we can see, okay, this is the data as it was read, and this is the data that was normalized. And if we run then the two final cells, then this saves two things. One, an Athena project created from this data that we read, and a metadata file in JSON that specifies what's the data contained in this first intermediate result, which is the process data, the collected data. This example took to run about 3 minutes, 4 minutes if we while we were talking. And we can already see the power of this because, for instance, the metadata, if you see here, it's pointing to patterns, not to file names, but to a pattern. And what it's doing is reading all the files with the same pattern and then merging them and then creating a new merge group in memory. So in reality, we in this short amount of time, we read 28 different files. They were merged according to the pattern that they display, and then they were assigned a name in this, which is going to be used in further steps of the exercise. And this is actually real data that we downloaded from the University of Southampton and corresponds to this publication from Veronica Celorgo and her colleagues. Andrea Russell. This is the first example, the first very, very tiny example of reproducibility. Okay, I think we can continue with the presentation then. So we have another 4 or 5 examples, but the idea is about the same. So we use metadata, we use the metadata to read the data into some variables in the notebook, and then we use the instructions, we follow the instructions one by one set on the second metadata file to reproduce the same results. Still, there is so that's why what we explained here, and we have another second set of examples. So we are going to launch a quick poll. Again, what we do is we go back to the browser, and we navigate using the browser to the place where we store locally these examples. So our example number 4 is the EXAFS example. It's EXAFS analysis. So in this case, while it's loading, I'll use tell you is a very all these the previous 2 intermediate two workflows did some processing of the data. So the SAINS analysis, which is not there is not much processing there. The linear combination fitting analysis, that's also just comparing the sum of 2 standards to see how they reflect the results of around the white lane of the signal. So in this case, we are going to do EXAFS analysis. And again, we have two files. If we go back to the directory, so this is this is a part of the Jupyter notebooks that lets us explore the directories. So this is the directory for the EXAFS analysis. So we can see the mapping metadata, which is just a JSON file with the names of the groups. What's the name of the project that contains this metadata, and what's the path to get to this project. In this case, this is an output because it's a result of previous processing during these exercises. And the operations metadata, which just tells us the variables and the different operations that we are going to be performing for the EXAFS analysis. And in this case, maybe you will tell me off for doing something that's very, I don't know if it's correct for EXAFS analysis, but let me show you the example and then we can talk later. So it's the same thing. First thing is we load the libraries. We wait for the libraries to load, and then once they are ready, then we can start running the reading of the data. So and the reading of the data entails to subprocesses or read the metadata and then get this metadata into the groups. In this case, the groups list is called SNKH data. So it has a number 1, so it means it's run. Obviously, in this case, I didn't clean or restart the Jupyter notebook. So you will see a number, and this number will change when I run it. So because it's a sequential number. So I executed this cell first, 2 I executed this second. So in sequence, this has a number 9, but now I expect it to change to 3 after it runs. So in this one, I'm just reading the data, and then I will try to plot it to see what we read from the examples. And the next thing is the reading of the fitting parameters. So that's also a bunch of data that's saved as. Oh, there is a question in the chat. Okay, yeah, the metadata JSON is the metadata, mapping metadata is being generated at the end of each workbook, each Jupyter notebook. So in this one, it's the last operation. Yeah, so if we go back to the previous example, the read metadata example, this save the Athena project only saves the Athena project, and save metadata, or save mappings, this saves the JSON metadata with the mappings for the what we just produced. The operations, that's a manual one, which we are just producing at the moment, but we are extracting that using another tool, which is called MLProvLab, and with another script that we have, but that's offline. That's not the part of the workflows yet. Where were we? Okay, so we read according to this, we run 1, 2, 3, 4, 5, and the next one would be this one. That's number 37. So in this one, we get the resources for the fit. In this case, the resources for the fit are the CIF files, the crystals, and we also need some information about where these crystals are located or obtained from, the absorbing atoms, the fit variables, the max radius to use, so that we can read and perform these operations. So we collect this data. Oh no, I don't want to restart that. So that's number 6. And the next thing is, before doing even the fitting, we do rebind the data, because these data sets are really big. So each one has more than 2,000 data points. So we do a rebind, which is also part of the normal processing. We rebind the data, and we save this data to an output file, which is listed here. So we did a rebinding of three groups: air, argon, and hydrogen. And after this, we then save the data. So we save this intermediate result, which is the metadata, and then we start reading the parameters for fitting. So in this case, we can modify or leave the fitting parameters as they are. So the first set of parameters is the guest-defined set parameters, which we use in Artemis as well. So we define the amplitude, the SSS, the E naught, and alpha for each of the paths. Then we can save those details, and then we have to generate the paths, the scattering paths running FEF on the list of crystals that we have. In this case, we have 4 crystals. So what this process is doing is running FEF 4 times. So we will see four times the outputs and the list of scattering paths generated by running FEF, which are here. This is one, this is the second list of FEF paths. And if we go back to the file explorer, we can even see where these files were generated here in this one. So each of these 1, 2, 3, 4 directories, they were generated by runFEF, and each of the directories contains the scattering paths for these crystals, and the ID corresponds to the number in the Cambridge crystallography database, because that's I just left the names of the C files as they were. So these are the 4 groups of scattering paths that were generated by this operation, which is the 10th operation. So the next one is the selection of the paths, and this is where maybe I did something wrong, because let me show you. I didn't select all the paths from a single crystal structure. Instead, I selected this path from the first crystal, this path from the second crystal, and this path from the third crystal, and I left this last crystal with no path selected. So I selected 3 paths for the fitting, and then oh no, I don't want to restart. And then when I run, I can see that these 3 paths that I selected, they are assigned some GDS parameters. So the delta R is varying according to this expression. We assign the sigma squared for each of the parameters, and they all have a common amplitude and E naught. So those are the values before actually running the fit. And this process only saves the parameters in case I changed something or added something here, and then we can run the fit. This is another good thing that you can see. So the 12, 13, this is going to become the 14 when it finishes running. And what this process is doing now is running the fit for the 3 samples selected. So for hydrogen, for the argon, and from the air sample, and comparing it to and trying to do a fit using the 3 crystals that I selected from these different structures, the crystal structures. And these are the results. So in each result, it gives me an R factor. So this is how good we think this is. And a fitting report along with the diagrams showing the fitting of the H2 rebind and its fitting result. So this is for the first sample. This is for the argon sample. I have the result, the R factor, and the fitting report. And this is the last example, which is the air sample and the fitting result. So, and it's the same. This is trying to replicate the results from the same paper that we started with in the example a few minutes ago. In theory, what I like to from this way of working is that once we have defined the metadata and the sets of operations, we can do this one-click reproducibility. So the thing is, if I clear the notebook, or I restart the kernel that clears all the outputs and restarts, and then I just click on run everything, which is this button with the double forward arrow, then the notebook runs automatically and produces the same results consistently. So in a sense, we have achieved the 3 lower levels of the pyramid that I showed before. We said repeatability, runability, and reproducibility. And I guess we could say that we also have some replicability, because this same structure can be used again and again with different samples and or different crystals. So we can start trying to mix and match or processing just with this simple tool. Still is a very underdeveloped tool, is still in under production, but we can start doing things like another idea that occurred to us during this research, that was, okay, what if I have many crystals or many path combinations and I want to test which is the best path combination? So this is the second example. I'm just talking you through it very quickly. So it basically reads the data from the Athena project. Then I give them a list of crystals. In this case, I just gave it a list of 1 crystal. But I told the program to look for the first 5 scattering paths and select the best combination of these 5 paths for fittings. So the results are these ones that I'm showing you here. So testing paths, the first only, and not the second, third, fourth, and fifth. So this has an R factor of 28.6, and this is the result. For the air milled sample, for the argon mill sample, we have using the same first path, we have this result, and for the sol gel sample, we have this result. And the program automatically does this testing, changing the combination of paths of first and the second, the first and the third, the first and the fourth, all of them. So in total, it does like 31 comparisons of these combinations. So we use combinatories to decide how many combinations, so that we tested everything. It runs in about 6 minutes, and at the end we can get a report saying which are the best path combinations. So for instance, the best combination for air milled is this one with 3 paths, the oxygen to Mn manganese, I think. Or for the second argon milled, this is the best combination that we found. And for the sol gel sample, this is the best path combination. So for the first one is 3, for the second one is 4 paths, and for the last one is only 3 paths. And we can even do it for even larger numbers of crystals, like testing with 5 or 6 crystals to see which crystals better reflect our sample. And this is an idea that I got from Khaled Mohammed. I don't know if he's in the chat, but he was giving me an example where he would need something similar to this just to speed up his analysis. We see that some examples, the R factor is terrible. For instance, this one where we only test part 1 and 2 is very, very bad. But we don't have to do them manually. So if this was like, if we were trying to test for 10 paths or for less paths, we can just let it run and just get these results. So back to the presentation. And I think I stopped it. So we already seen all these. Yep. So let me go to the questions. Amorphous materials. The short question, the short answer, I really not an expert on EXAFS processing. I'm more an expert on data management and programming. If you can deal with amorphous materials in Demeter and Athena, then it's just a matter of looking at the documentation and replicating the experiment. We can do it. That's the way we proceeded with the examples we have here. I mean, from the get-go, I didn't know how to do any of this. I produce these examples by reading the paper, reading the manual for the software, and coming together with these scripts that reproduce the same results as the paper. If there is a set of instructions for dealing with amorphous materials, so I guess it's when you don't have a specific crystal, then something like this happens. But maybe that's something like what I did with choosing different paths from crystals which are unrelated. They are already public. I mean, these are just Jupyter notebooks. They are freely available on the GitHub. The notes have the links to these Jupyter notebooks. There is you can see these examples that we were talking about and other examples. Plus, really, I encourage you to get in touch with us and ask us, and we can work with you, sit down with you, and try to see if the examples are useful for you, or if you have more examples, more ideas on what to do. Go back to the presentation. So these are the examples we have. And again, we started with this big workflow that we have the idea, okay, this is the EXAFS workflow. This is what we have to do. Read the raw data, produce the text files, read the data in Athena and produce the Athena project, and then use Artemis to do some fitting. But by reading the papers, we became aware that sometimes you start from Athena files, from project files, not from raw data. And other times you also add sameness analysis or linear combination fitted analysis to your data. And it's the same, you can start from raw data, Nexus or text, and you can also start from project data. So in this case, we already see the possibilities of building good set of tools that can be used scripted so that you can automate some of the processing and some of the analysis. And we're still thinking, as part of the PSDI project, that there is yet a better way. So the Galaxy project aims to create these tools which can facilitate automating even more. So in this case, we are creating a collection of Galaxy components that can be called from a simple interface. So this is an example of the interface we are creating. So in the left hand, you see all the tools which have the names of the processes that we want to perform. Then if you select one of these processes, the middle panel will show the a form in which you can input the parameters and the data that you are going to use with this tool. And then if you click the run button, then your results are displayed on the history. So you can, in a single interface, have all this processing power. Plus, in addition to this, you can then, after you finish a long analysis, extract the steps automatically and create a workflow which can be executed n times with different, varying the inputs, varying the parameters for different purposes. This is already tried and tested in other areas. The one that's more classic example is bioinformatics, but they have been used in astronomy, in biology, in biodiversity studies, and so this is just easy step from, and actually these tools are coded also in Python, so it's just wrapping tools in Python and exposing them as tools in the Galaxy interface, and then you will have these facilities. Very sorry, but I couldn't create an example for this presentation. But if you feel like it, the next time we can schedule a demo with the team from the scientific computing department, in which they can show us the same processes that I did here and many more, but with a simple Galaxy workflow that runs even faster because you can run it on the computer cluster. Variants of the fits. Well, that's again something that I cannot answer as an expert on EXAFS analysis. So for me, when visually, for me this is a bad fit because doesn't represent the first hump, and its R factor is high. So all this with a high R factor, for me, they are bad fits. And the better fits are, for instance, like this one, that almost all the peaks match each other in both the K and the R diagrams, and so that's how I do it. I hope that's but if there is other criteria, I mean, maybe, and if there is a way of encoding this criteria and reading it, for instance, from the fitting reports, so kind of like if because I don't know if maybe this what John is asking us is maybe in this report, so the statistics like the chi-square or the R factor. So I used R factor because I think my colleagues mentioned that that's very easy to see. Other things they said is, for instance, if you look at the S-square results, if you get a negative results, that's a no-no. That's something that shouldn't happen. So, but I'm not sure. So coming back to this Galaxy idea, the tools in Galaxy would be exactly the same as the ones that we are discussing here for the notebooks. It has many more benefits apart from being able to analyze data. You can work in an online platform, so you don't need to set up anything, to install anything in your computer. You can support common processing tasks. It's accessible for novice users and for expert users, so it's very flexible. It also supports the processing of very, very large data sets, because you can just let it run and just come back for your results later. So it's an ideal tool, especially in the view of improved beamlines or larger experiments of analyzing operando data. So I just want to mention the people that's working with us in the PSDI project, which is Leandro Liborio from the computational physics group, Alejandra Gonzalez and Patrick Austin from the data and software engineering group. And and this is a PSDI collaboration, but also informs other projects like the Euroscience Gateway and the Galaxy Europe project, where we are exploiting some of the results that they got for a similar tool for muons. And I also like to thank my colleagues at the Catalysis Hub, because they gave me many of the ideas about the analysis to perform, about what's interesting about them, and especially the group on X-ray absorption spectroscopy, Donato, who is now working with Diamond, and Jesse, and Andy Bill, and June, Santosh, all the guys there. So I think that's it for now. We are just in time for closing remark. If you have more questions, please get in touch. You can get in touch with the project coordinator or with me, and we are happy to help you and see if we can work together.
Thank you very much, Ebs. There is just one, I think, final question, which potentially you could either answer very quickly or in the chat. Just asking, do you have something similar for XPS data analysis at the moment?
No, we are looking into extending. So the first thing is we want to round up this and provide some tool for X-ray absorption spectroscopy. But we would also be very happy to work on other things like XPS, like neutron scattering, like Raman spectroscopy. So we don't want to limit this, because PSDI is going to attend to the needs of all the facilities. And so beyond Diamond, we have ISIS, we have the Central Laser Facility. So yes, I have had this question many times, and it's the same. If you already know of some, we can work. If there is the interest, we can work with the group researcher or group of researchers needing that.
Very quick little wrap up. So thank you very much, Abraham, for a great webinar. We'll definitely have to get you back again to do some more examples with some Galaxy workflows in the future. So hopefully we'll have something about that probably in the new year or something. As Abraham said, you can always get in contact with us. One of the best ways to do this is via our website. We have a contact form on there, or also you can find some of the members of staff that are working on the project and contact them directly. We're also on most of the social media media platforms such as Twitter and LinkedIn, and we have a YouTube channel where recordings such as the recording of this webinar and our previous webinars will be posted, and you can also join our JISC mailing list if you want to keep up to date when we send out newsletters about what we're up to. But I just want to say again a massive thank you to Abraham for a lovely webinar.